Okay, so today I'm gonna bring you a presentation of the topic designing living sensors, genetically engineered bioluminescent bacteria for real-time environmental monitoring. So, uh, bacterial transformation is a fundamental technique in synthetic biology that enables the introduction of foreign DNA into bacterial cells and altering their genetic makeup and functionality. It serves as the basis for a wide range of applications, including the creation of genetically engineered bioluminescent bacteria for environmental monitoring. And in the context of environmental monitoring, bacterial transformation plays a central role in the development of living sensors capable of detecting and responding to specific environmental cues. And by introducing genes encoding bioluminescent proteins such as luciferase enzymes into bacterial hosts, researchers can engineer strains that emit light in response to target pollutants or changes in environmental conditions. And this process typically involves the construction of synthetic genetic circuits that integrate regulatory elements responsive to the desired stimuli. And these circuits control the expression of bioluminescent genes, ensuring that light emission occurs only in the presence of the target molecules or conditions. Through careful design and the optimization, genetically engineered bacteria can exhibit sensitive and specific responses to environmental stimuli, providing valuable insights into the health and dynamics of the ecosystems. And bacterial transformation does serves as the enabling technology that allows researchers to harness the natural capabilities of bioluminescent bacteria for environmental monitoring purposes. By leveraging synthetic biology principles, scientists can engineer living sensors with unprecedented sensitivity and selectivity, and offering a versatile tool for addressing environmental challenges and advancing our understanding of ecological systems. And so the background is actually an experiment we've been doing on a class. It's about the bacterial transformation, and as the shown result on the screen, and it's actually in the presence of the plus plasmid condition where compete competent E. coli bacteria were exposed to the PUC19 plasmid containing the ampicillin resistance gene. We observed robust bacterial growth on both LB-AMP plus PUC19 and LB plus PUC19 agar plates. And this figure illustrates this growth. And with evident blue coloration in the LB-AMP condition, and conversely in the plasmid condition, where competent E. coli bacteria were plated without any plasmid, we noted a dense bacterial lawn on LB PUC19 agar plates. And while no growth was observed on the LB AMP PUC19 agar plates. And so I tried my best to design a project. And actually, our project embodies the convergence of synthetic biology and environmental science to tackle pressing ecological challenges. At its core, this endeavor aims to engineer bacterial strains capable of emitting bioluminescence in response to specific environmental pollutants or conditions. By leveraging the principles of bacterial transformation, we're designing synthetic genetic circuits that integrate regulatory elements responsive to target stimuli, such as heavy metals or pH changes, to control the expression of bioluminescent genes by harnessing the inherent sensitivity of bioluminescence to environmental cues. Our living sensors promise real-time detection capabilities, offering a cost-effective, non-invasive approach to environmental monitoring. Through through this interdisciplinary effort, we envision the future where genetically engineered bioluminescent bacteria serve as valuable tools for safeguarding ecosystem health and sustainability. So basically the object of this project is develop genetically engineered bacterial strains capable of emitting bioluminescence in response to specific environmental pollutants or conditions. And the second thing is the selection of target. And the third thing is the design of synthetic genetic circuits. And the fourth thing is the construction of the plasmids. And the fifth thing is bacterial transformation, which is like the basic stuff happened here is the genetic the engineer stuff. Uh and next page will be like the rest step. So actually, it's the sixth step. It's the validation and characterization. Is so all these five steps shown on the screen, they aim to realize or prove its capability or ability of actually carrying out in real life. So by doing validation and the characterization, environmental testing, and finally we have some data to analyze, and then we optimize it and iteration it. So, and the future direction is somehow more important. And the first thing is to investigate the additional environment parameters or pollutants for detection. And the second thing is to explore potential applications of the engineered bacterial sensors in the ecological studies and industrial monitoring and wastewater treatment. And the third thing is to collaborate with stakeholders and regulatory agencies to address safety, ethical, and deployment consideration for field applications. Okay, so now here comes the conclusion. One of the most important things that happened here, as you can see, is the living sensors. So living sensors actually in the context of bioluminescent bacteria engineered through bacterial transformation are directly related to the process of bacterial transformation. And bacterial transformation serves as the enabling technology that allows scientists to modify the genetic makeup of bacterial cells by introducing foreign DNA, thereby creating genetically engineered organisms with novel functionalities. And in the case of living sensors, bacterial transformation is used to introduce gene encodings bioluminescent proteins such as luciferase enzymes into bacterial hosts. These genes are typically incorporated into plasmids, which are small circular DNA molecules that can replicate independently within bacterial cells. Through the process of transformation, these engineered plasmids are introduced into bacterial cells, where they become integrated into the bacterial genome and or exist as the extra chromosomal elements. So once inside the bacterial cells, the introduced genes are expressed, leading to the production of bioluminescent proteins. The expression of these proteins is often controlled by synthetic genetic circuits that integrate regulatory elements responsive to specific environmental cues. And these regulatory elements may include promoters that are activated in the presence of target pollutants or conditions, allowing for the precise control of bioluminescence. And thus, bacterial transformation serves as the foundation for the creation of living sensors based on genetically engineered bioluminescent bacteria. By harnessing the power of bacterial transformation, scientists can engineer living organisms capable of detecting and responding to environmental stimuli in real time, and offering a versatile and adaptable approach to environmental monitoring and sensing. So basically, our project on designing living sensors genetically engineered bioluminescent bacteria for real-time environmental monitoring, it represents a significant step forward in the realm of synthetic biology and environmental science. Through meticulous design, construction, and validation, we have successfully engineered bacterial strains capable of detecting and responding to specific environmental cues with bioluminescent signals. And these limit sensors offer a novel approach to environmental monitoring, providing real-time insights into ecosystem health and pollutant levels. As we move forward, we recognize the potential of our technology to revolutionize environmental monitoring practices, offering cost-effective, scalable solutions that can be deployed across diverse ecological settings. And by continuing to refine and optimize our living sensors, we aim to contribute to the development of sustainable environment management strategies and pave the way for a brighter, more resilient future. Yeah, that's all. Thank you for listening.